Hello, good afternoon. My name is Koenraad Vees. Welcome to the poster presentation session. In this session, we will offer the opportunity to our research colleagues to present their work and to explain their work in the most non-scientific way to our family audience. So this is the challenge, and the winner will be announced tomorrow evening and will be awarded a €200 bursary and the prize of CNE. First, we will start with Professor Julian Midgley. Professor Midgley is a professor at the Department of Pediatrics at the University of Calgary, and he will be presenting two abstracts. First one is entitled Low Grip Strength Correlates with Low Bone Mineral Density in Cystinosis Myopathy. And the second one is on 2 successive pregnancies for a mother with cystinosis and maintained kidney function, the effects on kidney function and proteinuria. The floor is yours.
Thanks very much indeed. And I'm not really a researcher. I look after a whole bunch of cystinosis patients, and I'm very pleased to present a little bit of data here. Is it good? Do you have slides for me? Oh, how about that? Okay, so we all know that myopathy occurs as a late manifestation of cystinosis, and myopathy reduces muscle strength. That means that weight-bearing exercises are less, and less weight-bearing exercise means that bone mineral density may be on the lower side. We can measure muscle strength with a dynamometer, and this is really just to look at the relationship of grip strength to bone mineral density. And so we've got 16 patients with 3 measurements of grip strength at a particular time, close-ish to bone mineral density, but unfortunately the forearm density, which is the one I decided to use because it's closest to the hand, only had 10 measurements. We use Z-scores, converting a measurement into a Z-score to compare across ages and across averages which change. And so the Z-score ranges from 0, which is an average measurement for a particular age in the population, and it goes up to +2 for the highest level and down to -2. And you can see on the graph of the bone mineral density that we have some lines which represent the average and the +2 and the -2. And so in these 16 individuals, the Z-scores range from -3.5 to nearly 0, so somewhat below the usual range with the average of -1.5. And the bone mineral density, the average is actually below the usual range at -2.2, going up to nearly 0, but all the way down to -4.7. So clearly lower in this population. And when we look at transplant status, the transplant Z-scores are lower for grip strength and bone mineral density. And this is probably because, at least in part, the grip strength Z-scores and bone mineral density Z-scores actually decrease with age, and the transplant patients are all older. And so this is a graph of the grip strength versus forearm bone mineral density. You can see there's a bit of a relationship. There's quite a lot of scatter, but lower bone mineral density and goes along with low grip strength. So that is the main finding. This is the box which shows the normal area for grip strength and bone mineral density, and you can see that at least half the individuals are outside that normal range. And so a few patients were studied. We haven't adjusted the Z-scores for short stature because there's a bias in short stature. And handedness needs to be verified because at least one patient I see has a grip strength in the left hand more than their right hand, so I ought to check on that. And so in individual with cystinosis, grip strength is below average, as is bone mineral density, and the two are correlated. And I kind of use this as a sort of competition when they come to clinic to see how good their grip strength is. So I'm hoping that they keep their muscle strength up and their bone mineral density. We check every year or two, depending, to try and make sure that they have good calcium in their bones. So that's my first abstract. Oh, a question? I don't know if anybody's got a microphone. Yeah. So the question is regarding prednisone and the use, because prednisone is associated with lower bone mass or bone mineral density. And certainly that's probably one of the factors why the transplant patients have a lower bone mineral density as well. Although when you look at transplant patients, they will often recover their Z scores after a while. And we haven't really got enough patients measurements at the moment to look at all the possible things which may be affecting this relationship. But I suspect that prednisone also has an effect on muscle strength as well. So it may be that the two prednisone affects both measurements.
And in that case, do you think that the recommendations for prednisone use in cystinosis patients should be different than those with the transplant who do not have cystinosis?
Yeah, so it's a good question whether or not we should be using less prednisone in cystinosis patients. I think that the most important thing is having a successful transplant, but there are certainly places which use lower prednisone or steroid regimens for transplant, and that could be a good reason. And I'm really driven by this because one of my oldest patients, who's 57, has got a very, very severe myopathy, and his upper limb strength is so poor he can't even hug his wife anymore. So I really want to try and see better care for muscle strength. Okay. Well, let's just get on to a bit of pregnancy then. So this is a very interesting individual who has had a couple of pregnancies, and there's been lots of publications of pregnancies in cystinosis, but most of these have been in individuals who've had transplants. But recently, there's been two publications, one from our group and another one from South Africa, reporting pregnancies in individuals who've had no renal replacement therapy. And this is a diagram from that first paper looking at the GFR and the dotted line, the kidney function. And this is from the beginning of pregnancy onwards. And you can see that it's fairly variable, and the GFR increases as it usually would during pregnancy. The GFR was a little lower at the end. And then the blue line, which is the total protein, goes up a lot during pregnancy and came back down after pregnancy, somewhat variable as protein to creatinine ratios are in the urine. But you can see that it settles out at a slightly higher level. And so she delivered a nice, healthy boy, and that was good. And then about 2 years after that, she had a second pregnancy, and again stopped histamine. The proteinuria rose, and we actually introduced histamine at about 30 weeks gestation. But I wanted to look at the change of GFR and the various urine proteins before and after each pregnancy, and those are the data. So around about 3 to 5 measurements, just collected for routine care before and after each pregnancy. And this is the averages. The dotted line at the top is the GFR, pre and post. You can see between the end of the first pregnancy and before the second pregnancy, the GFR declined a little bit more. The proteins in the solid lines remained roughly the same. There wasn't much improvement. But before and after the second pregnancy, the GFR declined quicker or more, and the proteins increased quite a bit more. So that's the graphical data. This is the table, and you can see that the GFR declined around about 7% before and after the first pregnancy, but much more after the second pregnancy, nearly 20%. And the protein levels, the alpha-1 microglobulin kind of reflects tubular proteinuria somewhat, but it's affected by glomerular leak of protein. But the total protein had increased roughly the same percentages before and after each pregnancy. And so this is an illustration of the sort of issues that you have with native kidneys with pregnancies. And so we know that pregnancy in general can affect kidney disease. When we just discontinue the cysteamine during the pregnancy, or at least during the first and second trimester, that means that the kidney is going to be affected more by cystinosis. And this stepwise decline in GFR, more so after the second pregnancy and associated with this proteinuria. But this family are delighted to have two very healthy boys, and I'm very pleased to report this data.
I think what you're presenting is really important, because I think the issue will become more frequent, won't it, as the time to end-stage kidney disease is pushed further out. So I think filling this important gap in knowledge is really important. What was the gap between the two pregnancies? And
2 years, 4 months.
Did either of them have preeclampsia?
No, she had some issues with a urinary tract infection and delivered a little prematurely for the first one, but no preeclampsia. Usual aspirin as a prophylaxis for that, and obviously stopped other medications that she shouldn't have been on during pregnancies. In our clinic, I continue to look after the adult patients with cystinosis, so we don't really transition them. So we've got quite a few 20, 30-year-olds still with native kidney function who may be having pregnancies. So I absolutely agree, it's going to be more important.
I was just hearing someone talk earlier and suggesting that some of the anti-rejection drugs after a kidney transplant can be dangerous for the fetus. What's the sort of normal protocol for something like that?
Well, you would discontinue the medications that you shouldn't be using, like mycophenolate, and substitute it with azathioprine. And other medications such as ACE inhibitors or angiotensin receptor blockers, which are often used for proteinuria, would also be discontinued as well. So she went to a high-risk obstetrician clinic for the supervision of her pregnancy, where all that was taken care of.
Thank you.
Just one quick supplementary, if I may. So one of the challenges of managing pregnancy in women with chronic kidney disease is that the physiology of pregnancy is completely different to outside of pregnancy. And acid base is a big issue during pregnancy. And these women already have a tendency to acidosis. How did pregnancy impact upon that?
She didn't actually have any problems with electrolyte issues. She's on relatively small amounts of bicarbonate supplement. Her diagnosis was made very early because of her sister. So she's actually on relatively few supplements. Only recently a little bit of phosphate, not very much bicarbonate, but actually quite a bit of potassium. But she didn't have any particular issues during the pregnancy.
Okay, we will now move on to the next two abstracts. Abstract number 3 and 4. These are abstracts prepared by Louise Médard. However, unfortunately, she's not able to be here. Therefore, Rick Geesbers who is a professor at the lab of where Louise Médard is conducting her PhD, the lab of molecular virology and gene therapy in KU Leuven. He will be presenting the next two abstracts entitled Residual Cysteine Transport Activity for Specific Infantile and Juvenile CTNS Mutations and Isogenic Human Cell Models to unravel the underlying mechanism of cystinosis myopathy. Just wanted to mention that these next two abstracts are not in the competition for the CNE Family Award for Research Communication, just as the previous two, but then from abstract number 5, all of the next abstracts will be in the competition.
So thank you. The first one, I will explain you briefly what we did, specifically looking into some CTNS missense mutations. So these are writing errors at a protein level in the CTNS protein. Oops. Okay, I know. This title comes with the next poster. So this slide goes with the myopathy part. So I will first explain the myopathy part. The myopathy part was a CTNS Ireland, Cystinosis Ireland and Cystinosis UK funded HRB-based project, as indicated there, on developing a model for cystinosis myopathy, because we're currently lacking a good animal model to study myopathy, and there are actually also not good human cell-based models. I'm myself not a muscle expert. I work on viral vectors and I build models. So that's why I collaborated with this person here, Murillo Sampaolesi, who is a Duchenne muscular dystrophy expert at the KU Leuven. And what we used is this cell line. It's a human immortalized cell line, a myoblast cell line, which are the precursor cells of muscle cells. The nice thing is these cells grow in, can be grown in a dish. And what is cool about these cells is we can grow them as a myoblast in dish. And if we deprive them of some nutrients, mainly here the serum, these cells will fuse to early myotubes and finally to late myotubes, which are the first steps in muscle development. So in this project, we aimed, first of all, to use the wild type cells, to use viral vector technology to delete the CTNS protein. And like that, we actually built a model which is wild type and which has the cystinosis at the CTNS mutation, complete deletion of the protein. And then in the next step, to grow myotubes for them, analyze them in a second work package, and finally also assess if the phenotype we identify here, whether we can revert it by reintroducing a functional version of the CTNS protein in these cells. And that's what we showed. I can show the more detailed data in the poster outside. So what we succeeded to do is to build that model, to analyze it. And we detected, currently we identified a group of about 30 proteins that are expressed at a lower level. We still have to study what these proteins do and why this happens. But the interesting thing, to me at least, is that when we add back the CTNS protein, a functional version of the protein, that these protein levels go up again to wild type levels. So this provides us now with at least a model. We still have to look into what these proteins do, how it works, what kind of pathways might be triggered. But that will be next steps to take. Are there any questions about that? I was so clear. Not? Anyway, I will be around, so pull my leg or my arm. Then, right? So the second poster is also data that Louise gathered. It's a study where I tried to develop, together with my student, to develop a good model. So what we do is we build a model, we knock out the CTNS protein, and for me it's really essential that the phenotypes we see, that I can revert them, like make them back to wild type by introducing a functional protein. It's also essential to me that if I introduce a non-functioning protein, that's my negative control, right? 6 years ago, we started, and I said the best negative control is a CTNS protein that has a mutation. Because in literature, all the CTNS proteins with a mutation, they're considered non-functional. So what we did is, like, these are all point mutations, so writing errors in the CTNS protein. So a protein is built of different pearls, so to say, in a huge necklace. And one of these pearls, there are 20 different types of pearls, amino acids. But sometimes one amino acid can be replaced by another one, and that we refer to as a missense mutation. And that can affect the function of your protein. And that was also published, and that's indicated here. And it's related with juvenile or with infantile mutations. So initially, we started cloning these mutations and testing them in, this is a cell line, a conditionally immortalized proximal tubal epithelial cell. It's a cell that is covering the epithelial of your proximal tubules in your kidney. And these cells are deprived of CTNS, so the protein is gone. And what we did is with viral vector technology, we brought in the mutated versions, these mutated versions in the cell, together with a wild type. And what we observed in that, and then we analyzed them here, and what we observed is if you have wild type cells with the CTNS or cells that lose CTNS, we know that cystine levels, what you see here is cystine levels, they go up, okay? And what we observed is that here, when I reintroduce the first 3 dots here, are data points when we reintroduce a functional CTNS. So we rescue the accumulation of cystine because it's a functional protein. But to our surprise, actually several point mutations also rescued the accumulation of cystine. They do not all do that. For example, this is a very small deletion, and here the N288K also didn't rescue completely to wild type levels. And more interesting, this is, for example, the G339R mutation. There we do not have any rescue at all. So for us, that was kind of confusing. Because, so it's clear that there are some point mutations in the CTNS protein that kill the transport activity completely, but there are also several mutations potentially. We only tested these very few that still can rescue the transport function, and that may open different treatment approaches. Are there any questions about that? Okay. Thank you very much.
Oh, sorry, Anne-Marie's got a question.
Thanks, Rick. So does that mean that we need to get all of the patients with cystinosis genotyped?
For me as a scientist, that would be very interesting. So what it's clear, for me, these mutations show that there's selectivity. What I'm currently looking for is actually cell lines with these particular mutations, which is very difficult. We just picked these because they were present in literature. And these particular mutations, honestly, I don't even know whether they're present in Europe, yes or no. But I think, especially keeping in the back of my mind how the research in cystic fibrosis evolved, I think there might, personally, I have the belief that also for cystinosis, different point mutations might result in different phenotypes. And knowing that information would at least allow us as well to stratify patients potentially better. It's difficult to say whether that's going to be the case, but I guess so based on these data. Okay.
Okay, we will now proceed to abstract number 5. So I would just like to remind you that from now on, all the consecutive abstracts are eligible for the CNE Family Award for Science Communication. Our next speaker is Annemarie de Vreugd, not Anne-Marie. She is a nurse practitioner in metabolic diseases in Radboud UMC Nijmegen with a special interest in the outpatient care for cystinosis patients. Her abstract is entitled Patient-Reported Experience Comparing Immediate Release versus Delayed Release Cysteamine.
Thank you. We compared the experience of patients with immediate and delayed release cysteamine by asking them to fill in a questionnaire, which was spread through the Dutch and Flemish cystinosis group. They reached out to 65 patients and or their parents, and we got a response rate of about 58%. So 38 patients filled in the questionnaire. The questionnaire was based on a 5-point scale, so they were asked questions about therapy adherence, burden of treatment, and also their experience with side effects and quality of life. The patients who switched from from immediate release to delayed release cysteamine reported a higher therapy adherence. And therapy adherence is a big concept, but in this concept it was the taking your medication the way it was prescribed. So the delayed release group used it in 93% of the cases as prescribed, versus 73% in the immediate release phase. There was also a small group of 8 patients who stayed on the Cystagon, so the immediate release cysteamine. There was also, with the delayed release cysteamine, less halitosis, less nausea, and they slept better, which can also be explained by not taking any medication in the night. We also looked at the positive health concept. It is a concept to look at the overall impact of a disease or of a treatment. And there we look at quality of life, but also bodily functioning. So how do you feel during the day with the disease and or treatment? And also meaningfulness. And that sounds like a heavy term, but it's also your zest for life, your willingness to participate in daily life or daily activities. And we saw that the scores there went from overall moderate to adequate to overall good and very good. So there was the group who scored good and very good grew by 40 to 60%. So there was a really big change in how they scored those aspects of life. So overall, there was a very positive experience with changing to the delayed release cysteamine. There were 3 patients who answered the questionnaire who switched back to Cystagon because of the side effects, mainly gastrointestinal problems. So one of the patients also had problems with their weight. So the weight kept again going down because they couldn't have enough nutrition. And the last thing, we didn't correlate these findings, so the patient experience, with any biochemical markers. So we didn't check the cysteine, it's purely the patient experience with the drug. Thank you.
Thank you so much. I'm wondering, would the public have access to the questionnaire to see what those questions look like? I'm kind of curious to the qualitative data.
I think the cystinosis group has the questionnaire, so I think, yeah, you can have a look at that.
So that one was in Dutch and it's been translated, and there's a flyer
Exactly.
outside for our English version because we're collecting the similar data. It's not quite exactly the same, but it's close.
Based on the same questionnaire, yeah.
Hi, I'm just wondering, were all the patients taking the medication orally, or did you differentiate anybody who was taking it through a peg?
No, it was all oral medication, yeah.
I was going to say thank you for doing this, because it's very useful data if you don't mind us using it for our application for the UK.
No, sure. Yeah.
Okay, we will now move on to, yes, applause. We'll now move on to abstract number 6. This is now Dr. Jesus Mateus Martin, who will be speaking. He is a senior scientist at the Health Research Institute of Santiago de Compostela in Spain, and he will be talking about alpha-2 macroglobulin is overexpressed in patients with cystinosis, implications for the discovery of new therapeutic targets.
Hello, everybody. I have to say that I'm pretty new in the field of cystinosis. What I have been doing for a long time is to use proteomics to study disease. So, what is proteomics? For those who are not familiar with the term, proteomics is a set of laboratory techniques that are very useful to deeply study, to deeply identify, and to deeply quantify the proteins that are differentially modulated in the disease. At the end, the disease comes from an absence of a protein, an excess of a protein, or a malfunction of a protein. So with proteomics, we can quantify these differences. So what we did at the beginning is to get some skin cells from patients with cystinosis and also from the parental unaffected progenitors. So we grow the cells in culture and we treat the cells with or without therapeutic-like concentrations of cysteamine for 2 days. Okay? So what we obtain is the proteomic signature of each condition. And what we saw, I only have this slide to support, but you have the data in the poster if you are interested. What we saw is that an expected accumulation in patients of a protein called alpha-2 macroglobulin, which is a key protein in processes such as monocyte differentiation, immune cell regulation, and also endocytosis. And now we know that these processes are deregulated in cystinosis. So what we thought is that perhaps this was suggesting that the immune cells were playing an important role in the disease. So we got blood from a second cohort. This is a clinical cohort from Barcelona. And again, we obtained the peripheral blood mononuclear cells, the cells responsible for the immune response, from patients and from unaffected progenitors. In this case, we were directly to try to validate the results of this protein, alpha-2 macroglobulin, using a target technique, using antibodies, what is called immunoblotting. And we validate the result that we obtain in the first cohort of skin cells. So our data suggests also is preliminary data because we, of course, are limited by the number of patients, and this should be, of course, corroborated also in an isogenic model of the disease and perhaps in the mice model. But what we are doing now is to collecting blood from more patients. We are expanding this cohort and with other hospitals from Spain. And we are doing the proteomic analysis in the PBM cells, in the immune cells. So we think that we will have much more biological significant informations in the immune cells. The idea in the future is also to look for small molecules that are able to change the cystinotic phenotype of the cells, okay? So we are also collaborating with a high throughput screening group in Spain. They have batteries of small molecules, but these molecules are repurposing drugs. They are drugs that are already approved for another diseases. So what we are trying to do is to find a drug that have passed the several steps, the several phases of the drug development to accelerate the possibility that one of these drugs reach the patients in shorter periods that you have to develop a drug from zero. So now we are in this part of work in progress. I'm open to any questions. Thank you.
Thank you, very interesting. So you are using these progenitors, or what kind of cells are these cells? So unaffected progenitors, what kind of cells are they? As a comparison in your second cohort.
In the second cohort.
Yeah, so what kind of progenitors is there?
We are getting the blood of the parents.
Yes.
And the blood of the patients. So we want to compare at the proteomic levels the difference between parents, unaffected parents, and patients, homozygous patients with cystinosis.
Okay. So these are monocytes of the patients compared with the monocytes of the parents.
Exactly.
Right. Okay. Thank you.
Okay, I think there's one more question over there.
Sorry, Jesus. Did mTOR pop up in your proteomics? Did mTOR pop up on your proteomics?
No, not in our skin cells proteomics analysis. No. Perhaps it's because the kind of cell line. Yeah. Thank you.
Okay, thank you so much. We'll move on to the next one. This is abstract number 7 from Dr. Jinhao Zhang. He's a PhD researcher at the Stem Cell Institute Leuven Laboratory of Translational Cardiomyology of Professor Sampaolesi. And he will be presenting on human-induced pluripotent stem cells-derived skeletal muscle model to investigate cystinosis myopathy.
Good afternoon, everyone. My name is Jinhao Zhang, and I'm a PhD student in the KU Leuven. And I joined the research team of Professor Marilio Sampaolesi last year, together with Professor Rick Gispers and Luis Midar, to work on the project about the cystinosis myopathy. Let me give you a brief introduction about what I was doing and what I will do. As you may know, that cystic myopathy causes a series of problems about the muscle system of our body, which contains 50 to 60% of our body mass. Thanks to the effective oral medication, histamine, and the kidney transplantation, the lifetime of patients has been greatly extended. However, as their age grows, a series of problems can occur in their 20s to 30s. Such as the muscle weakness and the reduction of muscle mass in the upper limbs and in the hands, especially the muscle behind their index finger and their thumb. This muscle is quite important because the thumb contains the about 40% of the hand function. And other severe muscle problems can also occur, like the difficulty swallowing and unable to breathe. When you look into a disease and you want to see what is going on behind it, it's an ideal choice to build a model to mimic the real situation of what is going on. However, the previous model about cystinosis myopathy can only mimic the very early stage of the muscle cell development. In other words, they may be not matured enough. It's maybe because that the muscle cells can only stay in the plate for several days, and which is quite a early stage, I would say. And for me, I would like to try 2 different methods to help the muscle cells to stay longer in the plate. The first one, I tried the new material called extracellular matrix, the ECM. We found that the ECM with muscle cells, the group can form a much larger, bigger, and wider myotubes compared to the normal group. And then we tried a new kind of medium to help these cells to stay much longer in the plate, at least 11 days post differentiation compared to the normal group, which is normally 5 to 6 days, which is 2 times longer than the normal groups. But there's still a lot of things that we need to do, like to verify if the cells with the new medium with ECM, are they really more matured than the old ones? So that's the next step. And we also plan to utilize the iPSCs and the 3D bioprinting techniques to finally achieve the goal of making a more matured model. And that's all. Thank you.
Okay, thank you, Jinhao. We will move on now to Dr. Oisin Kavanagh from Newcastle University. He's a pharmacist and senior lecturer in pharmaceutics at the School of Pharmacy of Newcastle University. And his research interests are centered around relationship between drug physical properties and their pharmacokinetics. He will be presenting cystine crystal growth and decay in the context of cystinosis pathogenesis and treatments.
Thanks. Let me see if I can put it on the first slide. Great. Okay, so I guess I spend most of my life looking down a microscope at crystals, so I'm a little bit boring, but I'll try to make this as exciting as possible. So my team at Newcastle have been thinking about and wondering about cystine crystals. And we know that cystine crystals are the main feature that causes all the problems in cystinosis. And you can see here one nice big cystine crystal on the bottom left. And thanks to Professor Michael Ward, you can see a reference down in the bottom left, he's proposed this model of cystine crystals, which looks a little bit like this. And you can see here that in that crystal on the left, you're looking at the model like this. And each of the different colored blocks that you can see on the model that I have in my hand represents an individual cystine molecule, okay? And they pack together 6 cystine molecules in one Lego block, okay? And these Lego blocks then grow just like Lego. You can imagine if I tried to stack these Lego blocks together, one on top of the other, it would be a bit awkward and they would fall over. But if you stack them like a matchstick, so side by side, you can imagine then that those crystals will grow, right, in that sidewards direction. And that's exactly what we see here. We see thin hexagonal plates that originate from this center, right, this center, and they grow out in all of those 6 directions, okay? Now, why is that important? Who cares, right? Who cares if the cystine crystals grow in a hexagonal shape? Well, what's important for me, at least as a crystal engineer, is to understand the chemistry at these different faces. So we already get a clue. These crystals grow fast in the sidewards direction. They're more reactive, right? The chemistry is more reactive in that sidewards direction versus that top face that we can see. It's a little bit less reactive on that face. And what that means then is we think about trying to dissolve these cystine crystals is that we have a slow unreactive face and a fast reacting face that's going to grow. And we would imagine that that fast reacting face is also going to dissolve the fastest as well, right? And that's what we see in the clinic in a high flow system. Okay, so high flow system like the kidney, Lots of urine being produced, lots of fluid, lots of flow, right? Great. So we see that edge dissolution, and we can see that up the top. That edge dissolution of the cystine crystal is what dominates in the high flow conditions. But we also know when we think about cystinosis that sometimes these crystals form, I feel here a big echo, sometimes these crystals form in conditions of low flow, like the eye, like the inside of a cell, like a blocked kidney, right? So in those conditions, you've got slow flow, low flow. And if you look closely then, when we change the flow of liquid that we're washing our cystine crystal, we can see then the predominance of these pits that dig into the slow phase, okay? So we're already getting some clues about how to modulate crystal growth, crystal dissolution, just by studying the crystals. And the next step then is to understand, can we modulate the chemistry or try to understand the chemistry of how the drugs that are used in cystinosis work? And so what I did is I picked a big batch of different molecules that are proposed to work and they're used in clinical trials in cystinosis, and I washed the cystine crystals with these drugs. And there's one mechanism that's reported, and there's a little bit more detail on the poster outside of how these things work. But actually, when I wash the cystine crystals with these drugs, they're no more effective than water at dissolving the cystine crystal. Now, we know they work. We know in the clinic that these molecules do prevent the condition from getting worse, but what they don't do is dissolve the cystine crystal. They act at a different part of the pathway. They actually stop cystine from being produced. So it's slightly different. Now, for most people that might not matter, but for a chemist and for someone that wants to design new ways to perhaps have top-down dissolution or more rapid dissolution, that distinction is important. I can imagine a clinical situation where a patient maybe who was misdiagnosed or diagnosed late, cystine crystals were able to form in that patient. Cysteamine isn't going to dissolve those crystals rapidly. And if we could invent a new therapeutic that could rapidly dissolve those cystine crystals, we feel that that could be of value. And this is why we're so interested in spending all our days looking at crystals down a microscope. So thank you. Thanks. I brought this all the way from Newcastle and I crushed it on the stage. So yeah, anyway, see ya. Bye.
Oisin, we have one question.
Try to run away. No escape. Sorry. Yes, please.
Thank you. It's very interesting. I
Yes.
think your approach is especially interesting for cystinuria because it's a disease of kidney stones.
Sure.
And so I wonder, because in cystinuria, cystine crystals and stones are formed in urine.
Yes.
And so I wonder whether you are testing your crystal growth just in which environment you are, because I think the environment, the pH, all kind of stuff can be very important. So how you are approaching that?
Yes, that's a really exciting question, actually. So I've looked at crystals of cystine, which, as I understand it, can form anywhere in the body, not necessarily the kidney, but I was just picking a high flow and low flow situation just for the example. What's really interesting is that if you take some cysteamine, put it into a jar and just let it dissolve, it forms a really high pH solution of about 9.2, right? So does bicarb, as you know. And we use those things in the clinic to try and flush out, especially for cystinuria, try and flush out those cystine crystals. But what I did then was I changed the pH of the cysteamine solution to about 7, and guess what? It stops working. So it works by a pH effect in that case, but that's not by this disulfide exchange mechanism, which people think is what's happening. What I think is that it works earlier in the pathway and reacts with cysteine, and stops cysteine from forming and then depositing on the crystal and growing more things. But yeah, really interesting question. And we made an artificial urine to try and explore it, and we would love to look at other systems as well.
So
Yes, please.
Yes, I mean, mine is not quite a question because I have an evidence that when I use cysteamine at least 30 days and after up to 90 days, but already after 30 days, I reduce the crystals in the cornea.
Yes.
So how does it work, this, if you explain only that
Yes.
you don't form?
Yeah. So what I think is happening is that the water from the body is dissolving the cystine. It's not cysteamine because, you know, we take cystine crystals, we put them on a microscope slide. Hey, and there's no other thing, right? It's pretty simple experiment. You've got water, you've got cystine crystals, and I am flooding the thing with cysteamine at pH 7. Nothing happens. Nothing happens. I do the same thing with water, and the PhD student, bless her, she took photographs for 12 hours of the cystine crystals dissolving in those two environments, and she saw no difference in rate. So I think what you might be seeing is that the cysteamine is stopping the production of more cystine, which enables the cystine that's there to dissolve.
Again, I will show you the pictures
Yes.
where I see a lot of crystals
Yes.
at day 1 or
Yes.
day 0, and after 30 days,
Yes.
no crystals in the
Yes.
same
Yes.
cornea.
Yes.
So
I guess the cystine is not being produced anymore, and so
that was clear.
yes,
it's not
yes,
produced
and so
anymore. But then-
I guess if it takes 30 days for crystals, really small crystals, to dissolve, I would suggest that that's maybe a natural process and not due to cysteamine, unless cystamine's changing the pH of the eye, as I've indicated. But the point I'm making is not that the cysteamine doesn't work. It obviously works. It's that it works by a slightly different mechanism than what we think. And for me, that's interesting because I want to engineer new strategies to dissolve crystals.
Yeah,
if that
I
makes
mean,
sense.
of course, it's not clear to me the pictures that you show.
No,
Because
I
it's
guess
your
I must
experience.
say I thought I was presenting my poster outside. You're more than welcome to come in, and we've published the
Yeah, yeah,
paper,
yeah.
and
Let's
you
go
can read all
through.
about
Yeah,
it. Thanks.
Okay, we will now move on to the final speaker, Dr. Sante Princiero Berlingherio. He's a postdoctoral research fellow in the Laboratory of Pediatric Nephrology in Amsterdam, and he will be presenting targeting oxidative stress-driven lipid peroxidation improves podocyte dysfunction in cystinosis. The floor is yours.
Thank you very much, Conrad. And thank you, the organizer and the patient, to be here. Since I always say you give us the energy to proceed in our work. And yes, I will try to explain my PhD work. And the title is also quite complicated sometimes also for me. So I will try to make it easier. So basically, imagine that you are walking in this nice suspension bridge. Do you like it? It's nice? What do you think? It's good. Huh? But what if I tell you that actually your safety depends on these wooden boards under your feet? Because in case, if they are not so strong, you can fall down in the water. And actually, we have a similar situation in the kidney. Because, yeah, this is our body, these are the kidney. And in the kidney, we have the glomerulus. Basically, the glomerulus is act as a filter. And we have these octopus-like cells, this one, that are called podocytes. And these cells basically wrap the capillaries like the wooden boards. And what they avoid? They avoid that high molecular weight proteins or other metabolites present in the capillaries, basically they fell down in the urine. But actually, what happened in cystinosis? So in healthy condition, high molecular weight proteins and these metabolites are retained in the capillaries. But in cystinosis, we have some problems. So this has changed the color, but it's fine. So basically, in cystinosis, these podocytes, they detach. You see, they go in our sea, in the urine. And this is a problem. And because this is one of the causes why patients, they need dialysis and transplantation. So in my project, what I would like to do is basically how to solve this problem. First, I have to understand why these podocytes basically they detach. And then how can deal with this problem? So we took like these podocytes from the urine of the patients, we put in culture, and here you can find like a picture of our podocytes. You see they look like a little octopus. And we culture these cystinosis podocytes. And then what we have done is to make some tests on these podocytes. So the first test that we have done in 2 patient cell lines is to check how adherent are these podocytes compared to the healthy control. As you can see on the cells on the surface of like a dish, you see that present decreased adhesion. So if present the decreased adhesion, they will detach. But also what we notice that they present increased permeability. So if the filter is not working, it's more permeable. And this is actually a problem. So how can we solve this issue here? And yeah, if we think about the bridge, yeah, the bridge unfortunately is broken. So, and there is high possibility that we can basically falling down into the river. So we study these podocytes, and actually what we notice that, yeah, actually is not working the slide, sorry. So we studied these podocytes, and what we notice that this is the structure of the cells, you know, and this organelle is called mitochondria. It's like basically the power station of the cells. We notice that in these mitochondria are impaired. And if you think about the power station, if the power station is not working very well, what happens, I think that you have experience in this, sometimes you can have some smoke. And this smoke for us is like the oxidative species that at high concentration that are very deleterious for the cells. So can basically be harmful for the cell itself. And what we measure is these oxidative species, as you can see, they are increased in the patient cell lines compared to the control. But these oxidative species can also attack the lipid membrane of our cystinotic podocytes, causing this phenomenon which is called lipid peroxidation. And lipid peroxidation is very harmful for the cells because the conformation of the membrane is changing. And as you can see, we found an increased, like this is the graph, we found increased lipid peroxidation in the 2 patient cell line compared to the control. And yeah, how can we solve this problem? So we have to do something. And what we have done is to use it like an antioxidant against like the increase in ROS, which is called mitotempo, or we can also act downstream with another like a molecule. This is an anti-lipid peroxidation agent dealing like downstream of the cascade. And as you can see, if we like give this compound mitotempo, we have an increase of podocytes adherent compared to the non-treated cystinotic podocytes. So the podocytes are working better. And also we have the same phenotype with the other compound, which is called the liproxatin 1. And as you can see, already liproxatin 1 alone is able to increase the number of adherent cells on the surface. And not only this, because the 2 compounds were able to decrease the permeability. So both mitotempo and liproxatin 1. So this means that the filter now is working better. And actually, we would like to confirm the results that basically we obtained in vitro, so in cell culture, using this animal model. This is a zebrafish larvae model, and as you can see, it's like quite different from the normal one because it's green. Yes, this zebrafish model is like genetically modified and normally is transparent, and it contains this green fluorescence protein. So if the filter is working, the green fluorescent protein is retained in the vasculature of the filter. And as you can see, this will result in an increased fluorescence in the eye. So there is like a green fluorescent protein circulating inside. But if the glomerulus, so the filter, is not working anymore, this green fluorescent protein is lost into the urine of the fish. And this will result in a decreased fluorescence in the eye of the fish. And this is actually what we have in cystinotic podocytes. But if we give to the cystinosis podocyte mitotempo in combination with cysteamine, see what happened? That the fluorescence came back to the healthy control. So what this means that we were able, with these drugs, but not only with mitotempo, but also with liproxetin 1, we have the same results. We were able to restore the glomerular filtration barrier of this poor fish. And now, basically, we were able to fix our bridge from this to that. And I hope that we will join a nice walk together outside. Thank you very much. If you have questions, don't be shy. Also here to learn some. Something not clear or... Okay, thank you very much.
Thank you, Sante. So we are now at the end of the poster presentations. So the families are now invited to vote. There will be a poll, a Menti poll launched via the following QR code that is now being presented. So families, please vote for the researcher that was most successful in presenting their work in a non-scientific way to a layman audience. So I will just remind you that you can vote for Annemarie de Vreugd, Jesus Matthäus, Jinhao Zhang, Oshin Kavanagh, and Sante Berlingerio. And you are not able to vote for Professor Milgley and Professor Gijsbers. While you are voting, I will also think most of you have been able to scan this QR code. I will just go to the final slide. I have been told that there is a survey questionnaire that is launched by the research group of Anoush Chauhan and Rachel Bishop that is on the eye disease and use of eye drops. That has been introduced earlier today, and tomorrow Rachel will also refer back to this questionnaire. So if you would be willing to check this questionnaire and answer, then you're very much welcome by using this QR code. I will go back now to the code of the vote, and tomorrow the results will be tomorrow, Saturday evening, will be communicated, and then we will know who will win the prize for the CNE Family Award for Best Research Communication. I think we can finish this session. I'm looking to Denise. I think we can wrap up and then end the day. Anne-Marie has a question.
Thank you. Sorry, I just wanted to ask a question while the nephrologists were here, because I was afraid they'll be gone tomorrow and we wouldn't get the chance. But it's a general question regarding cysteamine therapy and dialysis, if that's okay. I want to ask, does dialysis reduce the effectiveness of cysteamine therapy, both cystagon and procysbi? I know it's different to what we've been talking about today, but I just, while we have the nephrologists, I wanted to ask.
Yes, I think it's a very important question, and there is actually no real data on that. We personally measured cysteamine in blood before and after dialysis, and also in the dialysate. It was done several years ago by Martina Bezau, and we also measured it in the peritoneal dialysis fluid, and we virtually found no cysteamine. So based on that, it was published. It was a letter somewhere published, but it was like measured in one patient. So So based on that, you would say that it's not dialyzed out. I know that there was a publication, actually the presentation by Greg Langman from Chicago. They did it as well, and they have shown, and there were different dialyzers, so it can be all different depending on the sort dialyzer, and they did not really show the clearance of cysteamine by dialysis, but they showed some difference in metabolism. But I think it will be good to make a study of that, to collect, to have, because there are not so many cystinosis patients on dialysis, but if we can really collect information in 5 to 10 patients and to measure cysteamine in the dialysis fluid, then we should be able to answer this question properly.
I'm sorry. So I was just asking, is there any difference in clearance from hemodialysis versus peritoneal?
Not that I know. Actually, what we have measured, but once again, it was one patient and one patient. So what does it say? The concentration of cysteamine in the dialysate in both, but of course the physiology is completely different. So yeah.
No more questions. Thank you very much. See each other tomorrow.